Great. Okay. Well, thank you so much for letting me speak to all of you. I'm super excited to share this work that I've been working on for quite some time. It's a behavioral project that I just recently released a preprint of. So here's the QR code if you wanna, if you haven't already taken a look at it, if you're curious. But the core kind of question that I'm looking to explore in my work is how sort of really rapid timescales affect our ability to form long-term memories. And so I hope I can encourage us to rethink the role that time plays in mnemonic operations. So the core question that my work is asking is, when are we best equipped to learn? So our intuitions might suggest that factors might influence our memory that evolve over minutes or seconds, like having a good night's sleep or a cup of coffee. And as a result, when we study memory in the lab, we often sort of assume that behavior unfolds over sort of multiple seconds. But remarkably, the brain points to a time scale that's too rapid for our conscious sort of apprehension, or I don't know, maybe I should be careful about saying that amongst a bunch of consciousness researchers, because maybe consciousness unfolds faster, but at least in terms of our own introspections, the brain can operate quite quickly. So there's a vast set of neurophysiological evidence and computational models that show that sort of our ability to form memories should probably actually fluctuate more rapidly than we previously assumed. And this is because the LFPs in hippocampus, when recorded from CA1 subfield, for instance, track moments of heightened synaptic plasticity, neural inhibition, and also perceptual drive. And all of these sort of ingredients are ideal for facilitating the formation of new memories. But after sort of 20 years of like rodent research, building up this sort of idea that and building this framework that would predict that memory should be able to be formed so rapidly, we still don't know if memory formation fluctuates on this rapid timescale. So is memory formation rhythmic? So to test this, we capitalized on the idea that attention-grabbing cues can reset the phase of ongoing neural oscillations. And so basically, if you were to just present an image without sort of any resetting of phase, you might expect that the pre-stimulus phase or the phase in the brain during this image might fluctuate randomly. And as you can see, it's kind of shifting. But if you present a cue before that image that systematically reorganizes neural phase, then the stimulus onset asynchrony between the cue and the target now tracks a putative neural phase. And capitalizing on this approach, previous work has shown that you can determine and quantify rhythmic states in attention. But this approach has not been applied to episodic memory, partly because I guess people might assume that maybe these effects are transient, they don't last, they're not persistent. And also, it's tricky to assume that people might be able to remember images that are presented very briefly, which is what this paradigm requires. So our paradigm adapted this approach for long-term memory. And basically, we present participants with a color cue for 100 milliseconds, followed by a stimulus onset asynchrony or delay between that cue and a subsequently presented object image. And if the cue is blue, participants are to make an indoor-outdoor judgment to that object image. And if the cue is orange, they make a size judgment to that object image. And critically, we then later test their associative memory for the object-task associations after roughly 2 to 4 minute delay. So really taxing long-term memory there, at least how it's done typically in the lab. And critically, we can evaluate how people's ability to form memories, the probability of later recall, fluctuates as a function of that cue to target stimulus onset asynchrony. And using this approach, we can determine if associative memory formation is indeed rhythmic, as would be predicted by these neurophysiological models and data. and we pre-registered our hypotheses as well, which you can see on the OSF. So to summarize our initial set of results, so to visualize how retrieval performance varies as a function of SOA, I'll first plot the raw average time series here in the upper left, where the encoding Q target stimulus onset asynchrony is on the x-axis and the hit rate is on the y. And as you can see, we see sort of prominent rhythms, in the associative memory retrieval performance when averaged as a function of the encoding SOA, and with about a 5% difference between the peaks and the troughs. So to characterize if these fluctuations are greater than would be observed by chance, we applied signal processing and permutation testing to each participant's time course. So this plot displays the FFT with the spectral power on the y-axis and frequency from 3 to 14 hertz on the x. And as you can see, we see the colored lines indicate the FFT from their memory performance, their memory time courses. And we see above chance fluctuations at 7 hertz in accuracy and inverse efficiency score, which is a combined metric of accuracy and RT. And we applied permutation testing to determine statistical significance by shuffling the SOA labels at the trial level and applied FDR correction to correct for multiple comparisons across frequencies. So, in addition, we then evaluated the degree to which these rhythms are transient. So basically, we used time frequency analysis to evaluate how long-lasting within the SOAs these rhythms are. And we found that these rhythms across participants persist for about 300 milliseconds or so. And this is just displaying IES here, but we have some other DVs in the manuscript. And we applied, again, permutation testing and cluster correction for multiple comparisons, and this is a significant cluster here. So an additional question is, we see sort of this phase synchrony in the group time course, which suggests, or rhythms in the group time course, which suggests that there could indeed actually be phase synchrony. So people could be better or worse at forming memories at the same times, basically. And so we computed the phase from the FFT and then evaluated its phase consistency across individuals. And we found, at least in IES, that the phases are consistent across individuals. So people are more efficient at encoding at a rhythmic level at the same sort of sub-second or millisecond moments. And so lastly, to get at this main question of like, are memories formed in a rhythmic way, we wanted to evaluate if they're formed rhythmically greater than would be expected, even if there was some sort of aperiodic structure in the surrogate data. And so we ran this control analysis, basically comparing the observed FFT spectra for each participant to surrogate data matched for autoregressive structure. And so each time course, for each time course, we generated an AR1 surrogate and then created a permutation distribution from those AR1 surrogates. But critically for this analysis, we averaged in the time domain. So this benefits from the phase synchrony as well, but it's a little bit more of a rigorous approach when considering the control, the surrogate data generation. And so as you can see, in all DVs here are significant after correcting for multiple comparisons. And this is likely also due to the fact that there's phase synchrony in both RT and accuracy, but it's sort of marginal. So taken together, what this shows is that our ability to form memories indeed fluctuates at a sub-second level, as long predicted by these long-standing models. But one remaining question is, to what extent might these rhythms in memory formation be driven by attention during the time of encoding? And so while we don't have a perfect metric of attention, we use the classification judgments during the time of encoding to ask that question. So I'll get into that now. So if you recall, during the time of encoding, participants also made classification judgments. So just to remind you, if the cube is blue, they made an indoor-outdoor judgment. So this, even though a plane is in a hangar, oftentimes, usually it's outside, right? So the correct response here would be outdoors, and we determined correctness from previous norming in our lab. So there's a little bit of ambiguity there. I kind of like this metric of attention because I think attention is in the real world is somewhat ambiguous. But anyway, what we first did is we just compared classification and memory IES because there's obviously this long-standing literature showing that, hey, attention and encoding should benefit later remembering. And when we look at IES, we see a strong correlation between individuals' classification IES and their memory IES, suggesting that there is in fact a relationship between memory and attention when you look at the aggregate. But next, what we really wanted to look at is these more rapid timescales. So there's a fair amount of literature at this point showing rhythmicity and attention, and so we wanted to see to what extent our data were driven by that. And so first, I'll just plot here the group time courses for inverse efficiency score for both memory and classification, and you can see that the rhythms here, at least visually, are more prominent for memory formation than they are for classification judgments. And what we can then do is evaluate the classification judgments statistically on their own to see if the classification judgments are rhythmic by themselves. And we apply the same methods that we use to the memory time courses and found no fluctuations that were above chance in their classification judgments in any of the frequency ranges. But there's still a possibility that the oscillations in memory are not statistically more prominent than those in classification. And so next, we compared their spectral power by taking the spectral power of the memory time courses and subtracting out the spectral power from the classification time courses for each participant, and then applying permutation testing to those comparisons by randomly shuffling which time course was classification or memory. And by doing this, we actually find that the rhythms in memory formation are more prominent. They're greater power than in classification for the inverse efficiency score. And the last possibility is that classification and memory could have common sort of phases, even if their oscillation strength is weaker. And so we compared the phase of the memory time courses to the classification time courses and found that there was no significant phase consistency between the memory rhythms and the classification rhythms, suggesting that, surprisingly, the memory rhythms we observe don't seem to be driven by attention during the time of encoding, at least on these really rapid timescales. And we were very surprised by this, but also I think there's some interesting future directions to explore when considering memory-attention interactions. So the last set of analyses, we were curious about sort of how putative cholinergic factors might modulate the ability to form memories rhythmically, because there's all of this literature showing that theta is in the hippocampus, at least, or in the brain, is modulated strongly by the acetylcholine. And so we used two analyses. One was related to sustained attention. So sustained attention is known to track sort of cholinergic tone. And so we quantified sustained attentional states during the time of encoding using the reaction time deviance. So this is an example from one participant for two blocks. And you can take the reaction time deviance on the preceding 3 trials, which previous work here by Megan de Bettencourt and from the Duncan lab by Lexi Decker has shown, who was my co-author on the preprint, that basically your RT deviance on the preceding 3 trials affects your ability to make attentional judgments and also for memories on the subsequent trials. So we applied that analysis to basically classify a putative high cholinergic tone and low cholinergic tone states. So here the green is low RT deviance, which is below that individual subject's median, and red is more deviant than the median. And so basically you'd interpret the in the zone states, or the optimal sustained attentional states, as the in the zone state. And just as you can see sort of visually, these blue dots indicate classification errors, and the red ones indicate subsequently forgotten associates. And roughly speaking, the errors seem to track these poor sustained attentional states. But if we look at it statistically, we can validate that our metric is indeed capturing what we think it's capturing we ran a mixed effects model and showed that when you're in the zone, you're better at classifying images, and you're also better at forming subsequently associative memories. And I later learned that this is the first, we actually were the first to show that this affecting associative memory, which is kind of cool, but here it's just our a sanity check. So the thing we're really interested in is, are people more rhythmic in their memory formation in these optimal sustained attentional states as compared to the poor ones? And so what we did is we sorted the trials based on their attentional state, and then we computed separate time courses as a function of SOA for the in-the-zone states and the out-of-the-zone states, with the idea that the in-the-zone states would reflect high cholinergic tone. And then we computed the spectral power difference between those two time courses. And here we found more prominent memory rhythms in associative memory reaction time for in-the-zone states as compared to out-of-the-zone states when applying permutation testing and correction for multiple comparisons. We didn't see it in accuracy or in IES. And if you have any questions, I can talk a little bit about that later. So we were really excited by this finding. One thing to note is that we did exclude participants from this analysis that had missing time points. So this is a subset, but the original sample was quite large. It was 125 individuals. So we had a fair amount of participants still left over. And then lastly, we wanted to more directly get at this relationship between cholinergic tone and memory rhythms. Some of the participants in our online sample reported a regular use of nicotine. And so we computed the individualized metric of their spectral power, and then evaluated using a GLM if nicotine users had weaker memory rhythms than non-users. And that's like direction one might predict, because regular use of nicotine can lead to receptor saturation and neuronal desensitization in individuals. And indeed, we find that nicotine users have weaker memory rhythms, this time in accuracy, than non-users. And to validate or to provide more confidence for this interpretation of desensitization, we then looked at time spent in the zone, so our other metric of sustained attention, but this time using the across-subject median. And we found that nicotine users spent marginally less time in the zone. So overall, they were in an optimal sustained attentional state less of the time. So that seems to support our interpretation that these chronic users have sort of less access to endogenously produced acetylcholine. So taken together, this links the memory rhythms we observe in behavior to things we would expect from the neural literature linking acetylcholine to theta. So I'll summarize our findings here. So we find that behavioral evidence that memory formation fluctuates across milliseconds at the theta frequency. And our work provides missing behavioral evidence for a long-standing theory for the role of theta phase in memory, which is called the Spear model, or separate phases for encoding and retrieval, which argues that during sort of specific phases, which it cites literature showing that at specific phases of hippocampal theta, there are specific synapses that are more active, specifically those allowing perceptual input in from entorhinal cortex to CA1 and CA3. And this theory also highlights that the reason why we have these rhythms is to differentiate trade-offs between encoding and retrieval. And we don't directly test the retrieval portion here. I can talk about some follow-ups we have to do that. But basically, the idea is that the reason why we might have these rapid rhythms is to facilitate interactions between perception and memory. And lastly, there's this really cool paper that inspired me a lot, showing that, basically postulating that we also have sort of these hierarchical mode switching events, basically. So on slower timescales, we switch between sort of an externally biased mode and an internally biased mode, but we also do the same thing on more rapid timescales, but in different on different scales, spatial scales in the brain. And what's really cool about our sustained attention data is I think it also provides evidence to support this idea, but in behavior. So there's these slow fluctuations that might be driven, perhaps sort of reflecting external versus internal orientation. And we have these faster fluctuations in internal and external orientation that are nested within sort of those slower ones. And this, I think, provides some really exciting and interesting inferences for just how cognition and the brain might work in general, and how cognitively the brain can enable sort of complex behavior by rapidly comparing perception and memory on multiple time scales to facilitate prediction and error-driven learning and things like that. So with that, I'll thank my supervisors, Catherine and Tofik Aliante, and my collaborators, Bjorn, Kay, and Chaim, and be happy to take any questions.
Thank you, Thomas. Bravo.
Thank you.
And, really nicely presented too. So thank you. I have lots of questions, but I will let the others start.
Sounds good.
Yeah.
I have one question actually about the very start of your presentation. So the whole research is based on this idea or this fact that these memory rhythms reset in the presence of some salient attention-grabbing stimuli. I'm wondering, like, if they how do the dynamics of this reset actually work? Do the rhythms reset at the onset of the stimuli, at the recognition of the stimulus, at the offset of the stimulus, and especially how does it interact when we show like the target stimulus afterwards, because that in itself is already again a salient stimulus that should also have an impact on subsequent rhythms, right?
Yes, it's a great question. So, I mean, I don't have great data to disambiguate all those possibilities, but I have some sort of intuitions. So, I mean, if you think about this sort of mechanism operating across the brain, there's lots of different things that could get reset. There's lots of different regions that could experience a reset, or networks, right? So I think of it as a relative thing, right? So for instance, let's just say the cue resets, the cue target relationship is relative to what's task relevant at the time, right? So for instance, you need to process the cue information to then make a judgment on the image, the correct judgment, right? So for instance, if they see a blue cue and they make a size judgment, that would be incorrect, right? So they need to kind of maintain that information and perhaps in working memory to correctly make the judgment, right? And so whatever reset is happening during the cue might be task relevant for the object, and whatever resetting is happening during the object might be task relevant for some sort of subsequent event, maybe the response they're making and things like that. So it's kind of like, I almost like to think of it as like a little dance, right? You have the leader and the follower, right? So presumably there's work showing that like phase resetting is kind of a mechanism for sort of neural communication more broadly. So it might be happening, simultaneous resets might happen in different regions at different times. And so I have some subsequent slides that I can show later for some follow-up experiments after you guys ask some questions, and we have some ways to try to get at that with the neural data to try to understand like what regions are getting phase reorganized and what consequences might that have for behavioral performance. Yeah.
Okay. Do you have more?
Just one quick follow-up. Just like one of the so thanks, first of all, thanks for the answer. I think that makes a lot of sense, that it would actually be like a more sophisticated process where we have like resetting of task relevant circuits, whatever. Because I was thinking about like whenever I hear about this like rhythmic, so to say, like these brain rhythms that are somehow associated with memory, my first intuition would be to construct an experiment that basically reproduces these rhythms, because your experiment is basically arrhythmical. Your experiment is just, it takes basically the time difference and then you imply the rhythm from that by basically the Fourier transform. So yep. Instead, like my idea would be, or probably there's already work done in that direction of like, for example, encoding sequences of like memorized sequences of images, of identities, whatever, and then either show them at the specified frequency or off the frequency, because this way one would maybe be able to more finely tune frequency against frequency, not time difference against frequency.
Yes, I agree. That is an ideal way of testing it. There are some practical challenges though. So this is not the only approach one can use to test rhythms in memory. So for instance, you could just look at pre-stimulus phase and how it affects performance. There's a fair amount of work on that, but again, you can't determine rhythmicity and behavior with that approach. There's also entrainment approaches, so you can like vary the opacity of a stimulus. So there's some really beautiful work, basically like varying the opacity of a stimulus and then varying the volume of a subsequent stimulus and then shifting the phase of the two of them to see if I'm forgetting the authors off the top of my head right now. But one limitation to that approach is you can't actually make predictions about phasic changes in performance, because it's basically you can determine if one rhythm is better than another rhythm, but not one phase is better than another phase, for instance. Now, with the idea that you had, you can look at phase, presumably, but the practical challenge you come across is how do you test memory for all of those items presented in encoding, and how do you present them rapidly enough so that they're actually remembered, and so you have to be slow enough so that they're remembered, but rapid enough so that you can sample all of those time points. So I've tried to think of a similar approach, but the challenge is like testing all of those items. But I do think that in some ways is a superior approach, but it's a little bit more practically challenging, I would say. So like one practical challenge we had was like the stimulus duration, so like 500 milliseconds versus less. And so, but one challenge you run into is like they actually need to be able to remember the images, right? So I think, and you make a great point that like we are looking across trials, and so it's not as ideal as one might like, but measuring behavior on sub-second timescales is hard, and that's why not a lot of people are doing it. But yeah, I think there's lots of other really cool ways to try to get at it, especially if you're interested in more ecological applications. So yeah.
Thanks for the answer.
Yeah.
I have a couple of questions as well. So thank you so much for the amazing talk, really clean and really nice results. So I have one question about so if I understood correctly, you showed that there is no rhythmicity in the classification part, right?
Yes. Yeah.
So I was wondering, how do you interpret that in terms of all this literature about attentional sampling and that is actually rhythmic? So is it because of the kind of task, or is it that the classification accuracy was already too high and then you cannot see these rhythmic fluctuations? Or would this attentional sampling be, I don't know, would it apply only to detection tasks and not to these more complex ones?
Yes, so that's a great question. So yes, so yeah, I mean it's a little bit hard to come up with a definitive answer, but I can kind of give you some speculation. So most of the papers that have looked at rhythmic attention look at basically most of them have looked at sort of simple geometric shapes and not like any complex stimuli with semantic information. I mean, there's a couple that have, but most of the prominent papers have not. And only a few of them have actually looked at non-spatial attention. So that's we're kind of in a rare sort of space. I mean, and a lot of the theories of rhythmic attention argue that the reason you even have rhythmic attention is for sort of sampling in space, right? So like you kind of the literature was started way back when there was this ongoing debate in perception about, can you sample things in parallel or do you sample things serially? And the reason why this literature was kind of born was that people were like, oh, well, maybe you're sampling serially, but you're doing it really fast and you're kind of shifting all around. So it looks like you're sampling in parallel, but you're actually sampling serially. And so that's one other reason our task is non-spatial. But there could be other sort of spaces, right? Like that you navigate that are like semantic space, right? So maybe rhythmic attention operates differently when you have sort of more complex multidimensional stimuli. And of course, the environment involves those type of stimuli on a day-to-day basis, and we use memory to guide attention in those circumstances. So I think my simple answer is just that we still don't really know how rhythmic attention operates in the non-spatial domain that well, and also how it operates in sort of semantic for stimuli with sort of all these semantic dimensions. And of course, you have to control for all these like factors. So we controlled for stimulus discriminability as a function of SOA, memorability, So there's all these complex stimulus dimensions that you have to consider carefully when designing these experiments. So it is a little bit challenging, but also you have to titrate the stimulus presentation. One thing I will show you though is that we did a control analysis to show that, to try to get at this idea of ceiling effects. So performance was higher in classification, but it doesn't seem like it's leading to less sensitivity. So basically, we looked at the proportion of SOAs that were at ceiling, and they're a little bit higher than the ones for associative memory. But when we look at the relationship between proportion of SOAs at ceiling, so the ones that were at 1, and the spectral power for each individual, there's no sort of negative relationship or no differential relationship between classification and memory. And this we seem to take as evidence that ceiling effects are not affecting our detection ability. Also, in some pilot experiments, we found alpha rhythms in classification, but across all experiments, we didn't find any theta rhythms. So, yeah. So we, yeah. No worries. Sorry, the Zoom lag. Thanks for the question.
So the other one is shorter. So it's about the nicotine association. So I don't know if I got it correctly. So it's more like a binary thing. Did they consume nicotine or not, or do you have some grading of how much?
Yes. Right. So we, yeah, I mean, I wish we had more information. We basically had this like pre-existing questionnaire that we use in the lab, just like, do you use or not? So it's not a very granular measure, but we have a follow-up experiment where I have an RA that's been working on an REB ethics approval now to do a nicotine gum or nicotine lozenge study. So we want to like basically give people that already use nicotine either like a nicotine lozenge or a placebo lozenge to see if it directly, and then we would predict in that case that it would enhance the memory rhythms in that regard. So hopefully that will more rigorously get at that question. Yeah.
Yeah, that's a really cool idea because in this case, I mean, you don't know really what's the cause and what is the effect. If you only have whether they consume or not, then you don't know really whether these people have already some other, I don't know, personality or other cognitive traits that would be associated to the memory thing. But actually manipulating the nicotine consume would be really a cool
So
idea.
what I will say, I didn't mention it, but we did actually have a bunch of other individual difference factors that we included in the GLM. So we regressed out like sex, cannabis use, psychological conditions, because we actually didn't exclude people based on the psychological conditions because their performance was identical to everyone else basically. And this was during COVID and online, so there's lots of people with conditions, so it would have excluded like a large portion of the sample that way. But that was our pre-registered exclusion criteria. And in fact, we did show actually modulation based on sort of conditions. So people with OCD had weaker oscillations, and people with PTSD had stronger oscillations. So this is accounting for all of that variance as well. But we didn't really highlight that in the paper because it was just like not enough space. But yeah, but we accounted for some individual different stuff as well, and this emerged even after accounting for that. So yeah.
Well, thank you.
Yeah. Thanks. I see Tian smiling.
Yes, because Lucia found the Canada PhD student payment rise.
And this is what Nature reports now.
Right, because in the past it was so bad. So hope, Thomas, you can get a raise for next year.
Wait, what's that, Tian?
I think it's so in the chat box, Lucia just posted the Nature coverage of how the SOS Save Our Science campaign succeeded and got passed by the Canadian Parliament, and students will get a better raise in Canada.
Oh, nice. Oh, yeah,
So,
right. Yeah.
Just for you to know, in Canada, we PhD students sort of try to comfort ourselves and say, "Oh, we get more done with less." This is sort of, the way we convince ourselves to stay in the Canadian academia, because it was just so bad. The research budget in Canada, drops by year instead of like getting a 20 or 30% increase as in other G7 countries. So, well, I'm very privileged to be in Europe. So.
So
Also, Thomas,
yes.
to me, this preprint is like my cousin. Like, it's not directly related to me, but I saw it grow and go from an idea to a manuscript to a preprint. I already know much about it, but your biggest fan today, Zohan, is not here. So I know he has some questions that he would like to ask you.
Okay.
First thing is, you're probably gonna mention it in too much detail, but these are all online experiments.
Yeah.
So the SOAs and the temporal synchrony is controlled remotely at the very beginning of the experiment design. So can you share some tricks on how to control, screen timing and stuff for online experiments and what platforms you were using?
Right. Yeah. So we used Inquisit, which basically the participants download and it runs locally on their machine. And then we presented the stimulus based on their frame rate. So basically we detected their frame rate once they downloaded, once they launched the experiment. And if it was not 60 Hertz, then we basically crashed the experiment and paid them some marginal fee for their original time. And then we reshuffled any skipped frame. If there was a skipped frame, we measured the actual time and reassigned a trial to a new SOA if it shifted farther than a given value. But the fluctuation in timing was only about roughly several milliseconds. So it was pretty precise. But yeah, and we used Inquisit, so Inquisit was actually quite good for the timing there. Yeah. Any other questions? I also have some other follow-up experiments that I can talk about too, if there's time. So I left them just in case there's some time. So
I have plenty. So if there is no from the guys there or anyone in the audience, I have plenty to ask you.
Okay.
I'm being patient, with them finishing theirs. No more
You
questions?
grill our speakers.
No? So I have so first of all, I think it is really cool that, you find these effects of fluctuating in the memory. Having said that, I'm still struggling with trying to understand the source of that phenomenon, right?
Yeah.
And the reason for that is because I think, and this is, I guess, what Jan was also hinting at. What we know is that attention is rhythmic, right?
Yeah.
And therefore, it's plausible that if attention is rhythmic, then of course it will have downstream effects on encoding, correct?
Yeah.
So you try to dissociate that between, say, like the classification task and the memory task. But the caveat there is that, say, in the case of the classification task, we know that, and this is one of the things that child differences has shown, is that if you are doing a task, you engage into this sustained attention mode, right? And when they find these wax and wanes, is more when it's not your primary task. So in essence, you can think that this dissociation that you're finding comes something to do with memory is irrelevant, right? You are not instructing your subjects to encode this. It's not that they have to do a task on the memory, right? You're just, later on evaluating the memory in and of itself, right? Whereas the task, it's the primary objective, right?
Classification task.
Exactly. So
Yes.
it would be great if you could have some other metric that could dissociate, that would actually have attention that is not task, right? Because now you're making the assumption is that I'm measuring that attention and task is one and the same, but it doesn't have to be, right? I mean, in principle, you could say your decision making is a continuous variable, right? And you're tracking that continuously, right? But the attention comes and goes, right? So if you could, eventually have something where these two variables are, where you could measure attention irrespective of a task, I think it would be much more convincing in saying these are not, the memory effect is not a downstream effect of an attentional effect.
Right. Yeah. I mean, no, I think that's a really interesting thing to point out and also an idea. I'm trying to think about how to measure. I mean, I know there's some ways in which people measure attention in EEG, I don't know, maybe mismatch negativity or something. I don't know.
Flash, you could do the things that I yelled at. So from time to time you just flash a stimulus and then you look for detection rates, right? But this is completely kind of task irrelevant in some sense. Like you tell your subjects that your primary task is to tell me whether this object is whatever, like you know, indoor or outdoor, or larger or smaller than a shoebox. And then say that you're going to have a secondary task that is not relevant whatsoever. We just, you know, like it's going to be there bothering you from time to time. But you know, like don't worry. Like if you can't do it, no harm, right? In that case, what they do, they prioritize. So they put all of the attention resources, you know, for the task on the kind of like, you know, primary task. And then for the second one, there's still like, you know, they did you could still measure attentional, you know, fluctuations, like I yell at that. And then you could say, well, if those, you know, because my prediction would be that there you are going to find wax and wanes.
Right.
Whereas in
I mean,
going to be sustained.
Yeah, one, I think that's a really interesting idea. One, I think practical challenge would be that, like, in order to measure memory formation rhythmically simultaneously while also doing that task, you would need to divide attention, because you would have to have the image, like, that you want them to encode presented roughly at basically at the same time as sort of some subsequent attentional decision. And so you might get like a memory that's at floor for that reason. And also they might also not even attend to the memory stimulus. I do think it's a really interesting idea. One thing I'll highlight, I was really wanted to include it, but I didn't include it. We had one experiment that we were debating whether or not to include in the preprint, which we decided at the end not to include because it's a little bit different in design. But we actually shortened the stimulus presentation to 400 milliseconds. And we had a backward mask because we wanted to like brief in the time that the stimulus is presented, because that's kind of like the ideal sort of design. And when we did that, we actually did see something close, like we saw almost above chance rhythms and classification in the theta range. However, when we did an individual difference analysis, theta power in the classification did not predict theta power in memory. And we actually saw stronger theta in memory. So that seems to suggest that it's not about detection, but there could just be separate rhythms that occur.
I think, you know, I'm not questioning your results, Thomas. I think that it could still be, you know, like rhythmic sampling, or I would like to believe that there's rhythmic sampling also for memory.
Right.
But, you know, from what we know, there's a lot of rhythmicity just because of, you know, like the wax and wanes of, you know, rhythms are not just in the hippocampus, unfortunately. There is much more rhythms for attentional fluctuations, you know, like, and those are, and if you could even just show that, okay, you still have a rhythm for attention, but it's different from the one in memory, it's even cooler, right?
Right. You know? Yeah.
So
I agree.
That actually would be, you know, even cooler, right? That's one suggestion. The next one is, I think that for the presentation of your results, you did not put, you know, or at least I did not see that you put a lot of emphasis on individual rhythms for different people, which would also be cool that you show, right? Because, you know, like you show like the wax and wanes is kind of like the group mean, but I'm pretty sure that some of your subjects, you know, like had, you know, like much larger wax and wanes, some were more like constant and so on. So it would be really great if you could show like, okay, what is the drift in terms of frequency, first of all, right? And also how many of your participants exhibited a rhythm versus didn't. Because at least in my experience, not everybody shows a rhythm. I'm sure that Urani knows more. Like she's the expert on oscillations in this stuff. And then you actually could have like a really neat separation, right, between people that actually have a rhythm versus people that don't. And you can go to your nicotine thing and even, you know, like say, okay, in those cases that are affected by these rhythms, I see a stronger, you know, the difference in the nicotine and in those that don't, for instance. Like that would be yet another thing to show.
Yes, definitely. We did look at that a little bit. We didn't include it in the preprint. It was like a long time ago. I did run like separate permutation tests for each individual, and I think roughly about 50% showed rhythms but we didn't correct for multiple comparisons because it's a little bit, there's not a lot of sensitivity. One of the reasons why we needed so many subjects was that we have to have double the trials of the attention task because we have to have encoding and retrieval. And so it's we're trial strapped basically. And so that's one challenge, but it is true. I actually did present. I apparently I was informed that Anthony Wagner's lab read my preprint, and I spoke to two of the postdocs from the lab afterwards, and one of them made the same comment, and he like ran this simulation, and he sent it to me showing, oh, you only need like 10% of these people to have rhythms to show it at the group level. And then but there is some variation in peak frequency. There's some variation in the degree to which they show rhythms. And I do think that would be great to characterize. So maybe your viewer will mention it.
It's not for your current papers, and your current paper is great, but then for your follow-up would be cool, right? Because also what I think is interesting is like you can ask in your follow-up experiment, what is the stability of that rhythm? So now you test those participants twice, and then you ask like, okay, is there any frequency drift? And then hopefully there isn't, because you are really tapping on something that is physiological, right? If it is physiological, then you wouldn't expect. So you would say like now the memory, like, and I think this would be your strongest way to demonstrate that this is there and it's functional, right? If you can take people twice and you say, okay, look, my peak frequency would be at 6 Hertz, but if you do it twice, I still keep in the same peak frequency, right?
Right.
You know what I mean? So not anything that you should do right now. Like I'm giving you more ideas for follow-ups.
Thank you. Okay. Yeah, I know. I think that's it's a really interesting idea. I'm really curious about the stability of the peak frequency as well.
Yeah.
It also is possible that the frequency might shift within individuals too for physiological reasons. But in theory, I could look at that too with my intracranial data because I have multiple sessions
There you go.
across different days, so we can see if it drifts within individuals as well.
Yeah. Yeah. So if you could look at that, I think that would be really cool to understand, like, is there something that, that makes, because ultimately if that frequency is there and, like your brain hasn't changed, even if it's not the same peak, the range should more or less be the same, meaning that, like if your peak frequency was at 6 Hertz, still like going now to 7 Hertz should be better than going to 8 Hertz. Right? Because, you're closer to your intrinsic, like peak frequency, right?
Right.
So there's some cool, like, and you can, perhaps you can actually team up with this postdoc because you can run cool simulations in terms of the kind of, because, you're trying to link this to the theta in the hippocampus. And as you know, the theta in the hippocampus has a frequency, right? So then you could link it to that, and it would be really cool to link it further, right? And last, did I see right that is it true that it seems to me that you have different amount of subjects in the nicotine group and in the non-nicotine group? Did I get that wrong?
Yeah. It's only, because we didn't, it wasn't like a priori prediction for us. So we weren't sure how many we were going to get. So it was an exploratory analysis. I think we had something like 25 out of 125. So it's a smaller group.
I would totally recommend you to, because as a reviewer, I would definitely link you on that. And I would say that, look, do me a subsample and show me the stability of that. Because if you have many more subjects, then it becomes a little bit unfair in the comparison of the sample, right?
Sure.
You could downsample the ones, I think that the one that did not have nicotine use were actually numerically larger than the ones that were in the nicotine group. And demonstrate that those differences that you observe are irrespective of. There you go. You see?
Yeah.
Because it could literally be that, like at least, when I saw these plots, I was like, well, these seem to be like more like random variations, and in one case you have more than the others, right? So
yeah. I mean, the reason, that's a great point. We acknowledge that. I mean, I know you're not trying to fundamentally criticize this. I mean, it is a, not the strongest evidence for this particular effect, and that's why we want to have a follow-up where we actually give people the lozenges. But I think that idea of subsampling the non-nicotine users to see like what percentage of the time do you see a difference when you subsample?
But I think in your case, you could, I'm not saying that this effect is not there. Like it could be there. Like if you demonstrate that it's there, like if you bootstrap and you compare with equal samples and it's still always there, I think nobody will contest that.
Yeah.
We'll have to believe you, right? You know what I mean? Like,
Right. Yeah.
Because why I nail you on that is because there is a possible explanation that
Right.
drives that effect that is simpler, which is okay, you have more people. And of course, more people have more variance, and more variance means, but if you subsample, then, and if the effect's still there, then fine.
Yeah, makes complete sense. I think it's a great.
I don't think they're or at least I wouldn't bother. I would stop there.
Yeah.
I wouldn't.
Yeah. I mean, one thing we're curious about too is like if we actually give people the lozenges, will it increase? I mean, because that's what you'd predict, right? So, we also, the design we have is to have 3 groups, like users that either use the nicotine lozenge or the placebo, and non-users to try to replicate this effect as well with a balanced sample size and things like that. Although we'll see if I have enough time to collect that before I finish up. But yeah.
But I don't think you need to. I think for now, like if you just do a bootstrapping, and you do the comparison multiple times, and you say, okay, look, out of 10,000 times that I do this, they can I downsample, and it's always there. Like fine, it's just there, no matter what, right? And this is more like a comment for all of you guys. This is a question. I know that oftentimes we do this analysis with like peaks and troughs, and and all that, but I'm always curious. Like, say, imagine that for a minute, and this is all also for you, Urani, with all of this oscillation stuff. Imagine for a minute that all of this is just regression to the mean. Like, how can we just separate oscillations versus a simple explanation of regression to the mean?
You mean that the phenomenon of neural oscillations themselves are?
Yeah, I mean, so you have a noisy variable.
Yeah.
So your estimate, you have a true estimate, which like let's say is 10, and you have noise, right? So that noise will adapt. Sometimes it will make it larger, and sometimes it will make it smaller. And that's the whole point of regression to the mean, right?
So shouldn't that be random? And why would it be rhythmic?
It will be, and what I'm just trying to say is that this randomness. So sometimes, like what we and this is exactly what I'm trying to say. Like sometimes we don't acknowledge that there could be just randomness. We just like make stuff like if it is in like what Thomas is doing now, right? If it is up, it's out, and if it's down, it's in. But what this is just like a regression to the mean. So then of course you're always going to have higher and always you're going to have lower. But this holds true for every measurement. I think that how do we define an oscillation? Up and down. But up and down is the same as regression to the mean.
Yes, well, so I will say, I mean, I think it's an interesting idea. I mean, maybe there is some truth to it. Maybe it's a certain type of regression to the mean, right? Because it's like the idea of oscillations is there's a balance between excitation and inhibition, right? And that sort of, if someone doesn't have that balance, they have a seizure, and if they, if they don't have any oscillations, then maybe, I don't know, they're just in a low arousal state or something. But basically, I have tried to look at these sustained attentional states, like using similar signal processing approaches, and they don't seem to be rhythmic. So they don't seem to be sort of prescriptively going up and down at specific times, right? So their structure is different. But I think you could frame both as sort of like sort of this idea of sort of balancing between extremes is kind of regression to the mean. Although you would expect it to then over time like go down, I guess, or approach the mean more, as opposed to have states where you have greater oscillations and states where you have less and states where you have greater. But like it's definitely hovering around some resting sort of state. It's some sort of homeostatic state of the system. But I think like you can quantify it differently by saying, hey, you're going up and down at a precise frequency, right? Versus sort of random sort of stochastic fluctuations. And typically, as far as like, I'm actually working with Lexi on trying to characterize like slower rhythms, like after errors, like across seconds. And at least some of the work I've done so far on this has shown that like these fluctuations that are slower don't seem to be strictly rhythmic in that regard. They kind of fluctuate up and down, but they're not at consistent times, basically. And the faster ones seem to be more prescriptively rhythmic.
That's why I'm asking you. I mean, this is not a question to you. This is more like a in general question, because I think that there is a lot of you know, like, I mean, look, I grew up in Webb Singer's lab, right? So the world was an oscillation for us all the time. But
Yeah.
over time, I came to also realize that even in gamma, it's really hard to have sustained oscillations, right? And in lower rhythm, it's even more. So there is this thing where you could say, well, what if it's not a true oscillation? It's just, you know, noise in the system.
Yeah, I mean, one of the things that really excited me about the Hasselblad model was like trying to understand like what's interesting about oscillations, because like a lot of people get excited like, oh, it's an oscillation, cool. Now we have some sort of theory. There's greater theta power for memory formation, and I'm gonna like call that a theory. I think the thing that got me excited about the Hasselblad model was like it's like, oh, okay, you have this like balance between excitation and inhibition, and you have very precise sort of functions that different types of rhythmic motifs can have. So there's a really cool paper by Olmsdorf and Tawfiq, I was the second author on it in Nature Neuro, where they had this review of like all these different types of motifs, and they looked at the single neuron level, showing like these types of neurons in this layer of cortex have this type of rhythmic motif, and there's like actually intrinsic oscillating frequencies of these neurons, and that their frequency can get modulated, and these sort of frequency modulations can perform a lot of different sort of computational operations like gain modulation or gating. And I think the thing that's really cool that I think is really cool about this idea of the Hasselblad model is this idea of gating, and like especially when considering perception and memory interactions, because when you look at the hippocampal circuit itself, the thing that I thought was really cool is that like, but you have multiple inputs going into CA1, right? It's like it almost feels like it can't work without oscillations, right? Like you get two inputs coming in simultaneously. What is CA1 going to do? It's gonna be like, I'm so confused. Like, why are two people talking to me at the same time? But like, it makes sense. Like, that's why I thought that this idea of like gating as a motif is such a interesting thing. And to me, it's more than just like, hey, there's a rhythm and there's not. It's that you have like prescriptive times when you're prioritizing certain information. And in the brain, there's all this evidence saying there's specific times when certain synapses are prioritized. And what we thought was powerful about this behavioral approach is we can say, hey, here's the behavioral correlate to that, right? There are certain moments when certain types of processing in memory is prioritized. And so we have a follow-up where we want to manipulate the SO8 retrieval and see if they're phase offset to really directly get at the Hasselblad model. And I have another follow-up, which I didn't get to talk about, which was about proactive interference that my RA Alan is running here, but I don't have time for that. But anyway, I guess the point being, like, I think the idea of I guess I would draw a distinction between like sort of the computational role of specific types of oscillations versus the observation of an oscillation at a network level, right? There seems to be a very prescriptive function that they might have on on multiple scales of analysis. But I also am quite skeptical about if they're epiphenomenal or what the role that they might have, which is also why I was super excited about the Hasselblad model, because I think it's like a way to try to describe to show how they might not be epiphenomenal. So yeah.
Here's a suggestion for you and Shen to think about. So one of the things that we do know is that eye movements activate the so-called like PGO waves. So eye movements create like a volley of acetylcholine, right? And they actually literally reset the alpha and create these so-called like PGO waves that are actually long-lasting. So one thing that you could do is that instead of having your reset as like the color, you could actually do ask people to do an eye movement and then flash the images. Right?
Yeah, so we have that. Yeah, we have that. So
right.
that was actually my first idea. So back during when I started during COVID, my original idea had nothing to do with rhythmic sampling. I was actually going to time lock everything to eye movements and then create an oscillating stimulus.
Right.
That was only I guess 2 milliseconds, and then shift the phase. And that would be the main manipulation. But I never ended up doing it because I had to run study online. So we pivoted to the behavioral oscillations approach because it could be done online. And I still never came back around to that. I actually have a version where I looked at eye tracking and have a longer Q duration, and I want to time lock the subsequent memory to saccade onset. I was going to include it, but I didn't have time. But in the intracranial data, I have like 6 patients so far, and half of, I would say 5 out of them, I have the second version where we have a longer cue that's either on the left or the right, and we want to time lock the subsequent memory to saccade onset. So that's building on Heim's work showing the phase reset in hippocampus with saccades. So yeah, we'll see how that works out. It's we'll see how sensitive it is, but yeah.
I put you in the chat the PGO waves. You know, that's something that you should definitely look into, because they are in the alpha rhythm, not in the theta rhythm.
Interesting.
Yeah, so look into that.
Yeah, I'll check it out. Thank you so much.
Cool. Those were my
And
questions.
I'll also say I didn't get a chance to mention the follow-ups, but obviously Tian has been incredibly helpful helping get my intracranial version up and running and with the analysis. So I want to send a major shout out and thanks to him. It wouldn't be possible without his help.
Always
So,
a
I'm sure you was. You're a good couple, guys. You know, a good team.
yeah. So
And I'm probably going to mention, but Thomas will be the guy who continue to collect data for me in Toronto. So big shout out to Thomas, my helpful friend. We actually just talked yesterday just to review how to run screening session and sort out the most selective images to be used. And yeah, and we're ready for the next patient.
yes. And so yeah, I'm excited to run Tian's task as well. I also did my spike sorting on my first patient last night. So, I mean, another cool thing I didn't get the chance to mention. One thing also, speaking to your thoughts about rhythms, Lucia, is looking at the single neuron level of analysis. So like we can look at broad versus narrow spiking neurons to see if they fire at different phases. I'm really curious to look at spike timing correlations as well as like a measure of plasticity. So like I had the thought to like select certain pairs that have higher spike timing correlations, and then see if those spike timing correlations fluctuate with theta phase, with both the behavioral and the LFP theta phase. And then I'm getting inspired by Tian to also look at use Zebra and see if there's like these state changes that also fluctuate with sustained attention or SOA and things like that. That might not strictly be like present in the LFP, but might be present in sort of this pattern of spiking activity. So there's also a possibility that it's not the LFP in hippocampus per se, but the phase synchrony of theta across networks. That'd be a little bit harder to get at with intracranial because we have different coverage for different patients. But there's a lot of ways in which this could map onto the brain that's not strictly just LFP in the hippocampus, which I think would be exciting to explore. But
here you have actually the eye movement data would be really cool, because the eye movement create this, phase reset all across the hierarchy, right? So then you would have a very nice way to look at, like you know that, right? So if you actually log to the start of the saccade, you have like an ERP that goes all the way from B1 to hippocampus, right?
Yep.
So you have the whole network kind of on the same clock, right?
Yeah. Well, I'll have to. I'm just using Tian's scripts to align my eye tracking times to the intracranial data. So thank you for contributing to M&E and also making all the great functions there. It makes it a lot easier to do those types of analyses. Yeah. Actually, what's that?
It's good to be a good citizen.
Yeah. Also, I'll do a little shout out. So I'm also for my outside project, I'm reanalyzing some of Chaim's stimulation data. So he actually capitalized on this phase-locked ERP to eye movements and then stimulated it at the peak or the trough. And I'm reanalyzing that to see how that affects eye movement dynamics, actually. And it seems like the stimulation at the peak seems to benefit visual search performance as well. So I'm in the process of now, like, I was working on that with Jen Ryan, and I wrote up my outside project. I'm going to be presenting that at SFN in Chicago in a poster. So I'm starting to look at the neural data with that too, but it's a lot of data cleaning as well. But that's kind of the flip side. It doesn't have to do with memory, although his task is a memory task, but I'm looking at the effect of hippocampus on eye movements for that one. Yeah. So. Yeah.
I'm done with my questions.
Okay. Good questions. Any other questions? Anyway, thank you so much for giving me the opportunity to present. I really enjoyed chatting and hearing all your questions and thoughts. So,
Good luck on publishing my cousin.
thank you. And hopefully the cousin's child. Well, I don't know how to refer to the intracranial project, but hopefully that will. I mean, that's going to take some time, but that will be fun to work on as well. But hopefully this gets in review first soon. Yeah.